All right, so we're discussing photosynthesis, right? So basically, guys, with you guys, just want to recap, not really recapping, but let's talk about Kelvin cycle. So I think we talked about what this cycle was, what the final product was, and it's just basically what's happening here. It's the overall process, and I kind of give you guys a breakdown of the three phases of the Kelvin cycle, right? So the first one, as we talked about, is carbon fixation, fixing carbon, right? And second was reduction, and third is regeneration. Okay, so I asked you to read the chapter, so hopefully you have a good understanding after when you guys lead to that. So the Kelvin cycle, as we know, it divides into three phases, right? So this is the first, this is the second, this is the third. Okay. So the fixation part, okay. So we talked about it, guys. I mean, we talked about it for the AP class, right? So the problem with the fixation is where it's CO2, guys. It's a relatively rare gas molecule, as we talked about, right? So 70%. I mean, you guys are coming from chem here, so 70% will be N2 gases, and like 20% is O2 gases now, and less than 1% is CO2, right? That's one of those stuff that we talked about. So basically, what happens is, so there is, so it's a cycle, okay? It's a cycle, as we know, right? Just like the Krebs cycle, it's a cycle, okay? So there's an enzyme that we call rubisco. So with the Olympiad, I think you might have to know what it stands for, just so you know. Okay, so with the rubisco, the enzyme, right? So there's ribulose, ribulose biphosphate, biphosphate glycerate. This right here. Let me say ribulose biphosphate. Yeah, that's what it is. Yeah, that's the name. Ribulose biphosphate is the name. So this is a 5-carbon molecule. Okay. So basically, what it is that the rubisco enzyme, where the ribulose biphosphate carbonates. So basically, what it does is it takes the CO2, okay, and then we'll put it onto this guy, okay? And then I don't know, like it should sound pretty similar to you guys. Okay, so what the product of the rubisco is when they fix it, which is a range of CO2, and binds to the ribulose biphosphate. So the resultant is phosphoglycerate. So right there. Yeah, so Andy, you tell me, right? Where have you seen this one? Phosphoglycerate? Andy? Where did this product come out from? 3 phosphoglycerate? Where did we talk about this one?
I don't remember.
Yeah. What about you?
Step 6 of the.
Huh?
Is it the Kelvin cycle, the citric acid cycle, step 6?
No. Well, yeah. What do you think? Where have you seen this guy? This is okay. So I'm on camera because I mean, so I don't want to be mean to you guys here, okay? This is from the glycolysis. Okay, this is from the glycolysis. So glycolysis, if you recall, was glucose, it was glucose, and then phosphofructose. Remember that, guys? It was biphosphofructose, and then they turn to G3P. Like you gotta know these guys, you know. And then they turn into what, guys? And then so like you'll see this, okay? There's energy investment, energy payoff, okay, in terms of the glycolysis process, right? This is what we talked about. So this is where, like, dude, I tell you, I can directly tell you, this is where the problem is for students. They don't recall. We talked about it last week, and you probably were thinking, like, what the hell? Like, what, when? So during this process, glucose went in, and then what happens, guys? I mean, G3P. This is the end of the energy investment, okay, which means, like, they have to, what, put ATP there. Okay, and then during the payoff phase, okay, which means like they're using the dehydrogenase and taking the electron out. I mean, taking the they're putting NAD plus there, taking the electron out from this one. Then you get what, guys? And then you get phosphoglycerate. Okay, that's what pretty much what you get from the energy payoff process. Okay, so and then like what was the next one, guys? They use the kinase, right? And then what, like, you know, like, yeah, it's a phosphoglycerate like here, and then this is a I think biphosphoglycerate, and then I think they have G3P. So basically what I'm saying is, I mean, this is basically, you've seen them under the glycolysis process. So what we're discussing is we are going backward. Okay, we are going backward here. Like we are starting from here and going back to here. So this is the final product of the Calvin cycle, and then they can modify this to glucose. Okay, that's basically understanding. Okay, so basically, keep in mind, guys, here. So this is a cycle, so which means if I'm not mistaken, guys, 3 of these guys go in. So overall process perspective, 3 of these guys go in. Okay, 3 CO2s being added in. Okay, just, you know, for the cycle perspective. So we have 15 carbons for 3 of the ribulose biphosphate, and 3 CO2 is added in. So I think we get 6 of these guys. Okay, just which is 18 carbons overall. Okay, so again, like that's what the ribisco enzyme. Okay. So this is what we call the ribulose biphosphate carboxylase. That's the enzyme. That's what this stands for. Okay? So it's a ribulose biphosphate carboxylase. Okay? That's the enzyme. So this is where I tell you, like I'm telling you, okay, dude, you have to memorize this. Okay? So sooner or later, guys, you guys have to memorize this. You know. And then basically the first thing is what? So what we talked about is what? So you're turning a CO2, and the final product is what, guys? I mean G3P. So you're turning a CO2 into G3P, which is what? Right here. Okay. So which means you have to add electrons and energy, okay? So you're giving it the electrons and also the energy. So the first process is they're giving them ATP. Okay, so ATP turns into ATP and phosphate. Okay, it's providing them the energy for this. And then the phosphate group is going to get on this one, so it turns into 1,3-biphosphoglycerate. So basically, after this process, it will turn into 1,3-biphosphoglycerate. So that's basically, guys, first thing first, what happens, guys, okay? When the ATP, okay? So basically, in terms of, we had a discussion where, okay, so where do they produce ATP, right? So where do they get that from? It's not mitochondria. Okay. And after this, guys, so which means the first thing first, we have to add 6 ATP there because there's 6 molecules here, 6 ATPs, 6 ADPs, and then 6 phosphate will be in here. So that's why we have a biphosphate now, right? But now keep in mind, guys. I mean, this is as we talked about. That's the using of the ATP fixation process. So what about, guys, the reduction process? Where do we go from there? So reduction is basically, reduction is giving the electron to this guy. So basically, what will happen is there is, you know, NADPH. So it's very similar to NADH, okay, but they have just one extra phosphate there, okay. So having one extra phosphate, I mean, doesn't make a main difference at all. So this is where they carry the electron to the Calvin cycle, which happens to stroma of the chloroplast, okay. And then they have 6 of these guys provide the electron to the 1,3-biphosphoglycerate, okay? And then the product is what, guys? If I'm not mistaken, the product is they produce 6 G3P. So this is a reduction process. A reduction, I mean, so I can really put it here. So this is where the reduction happens, okay? And then the product is 6 G3P. So this is a glyceraldehyde 3-phosphate. That's what we talked about, okay? So which is right there. That's the glycolysis. So with this, again, with the G3P, okay, what happens, guys? Yeah, dude, they can make that into glucose, guys. So they just have to backtrack, okay, going into G3P to glucose. It's like what, 3-4 step process to make them into glucose, okay? Or they can take the G3P and what, produce, you know, fatty acids or whatever, okay? They can produce whatever they want. So that's the process. So basically, what will happen is, keep in mind, guys, we put 3 CO2s, we get 1 G3P. G3P, glyceraldehyde 3-phosphate, that is 3 carbon molecule, okay? So only 1 guy goes out. So keep in mind, guys, 6 ATPs, okay? 6 NADPHs, okay? For what? 1 G3P. It's a lot of electron needed, 12 electrons, okay? So like if you're smart guys, like keep in mind, okay? So we are putting in what? So carbon has charge of +4, okay? That's total of what? Total charge of what? +12 for three carbons here. So how many electrons do you need? 12 electrons. That's why we're putting 6 of these guys. There are 2 electrons per. You know what I'm saying here? So, and then what happens, guys? And then 5 G3P will be used to what? To make sure that what? This is the process of regeneration, okay? So 5 of these guys, so we produce 6, okay? Only one of them will be going out in what? 5 of them will be going back to the cycle for the reproduction of the ribulose biphosphate, okay? This happens at the expense of ATP, okay? Where this takes what, at 3 ATP, I think. I guarantee you guys, your teacher will not talk about this. Say, oh yeah, there's a Calvin cycle, okay? And yeah, good luck, guys. You get produced sugar next chapter. That's what they're gonna do, but we can't do that, okay? So that's the regeneration portion of it, okay? It happens at the expense of ATP. So meaning to produce 1 G3P, which is a glycerol 3-phosphate, you have to put in 9 ATPs and 6 NADPHs, okay? So the real question becomes, where are they gonna get this from, right? That's what basically where we ended up or whatever we talked about it. So, but before we go there, okay, before we get to the that portion there, first, if there's any question, let me know. Any question here? Some of the stuff we talked about, guys. I don't know when, but any questions here? Let me know. So one of the things, guys, that I can't really call this an issue, guys, was what? The problem with the Rubisco. So if you've done the reading, guys, the reading kind of mentions Rubisco, and as I mentioned this a few times, is the most abundant enzyme that's out there. Okay, that's one of the stuff that we talked about. Every plant or any photosynthetic, organism have Rubisco. Okay. So, but the problem is, when temperature gets a little hotter, as per our discussion, what happens, guys? They start to build the affinity for the O2 gases. So instead of bringing a CO2, what happens, guys? They bring an O2 gas. That is a big problem, guys, with these plants. Okay, because why? They need a CO2 to run this process. Okay, so the stuff that they added, which is an NADPH and also ATP, they got it to when they fix the carbon properly and able to add these things in there and be able to produce G3P. But the problem is when the Rubisco temperature gets a little hotter, we talked about it, guys, the shape of the enzyme changes a little bit. They start to bring on O2 gases. So it builds the affinity for the O2 gases. So what you guys have to know is what the hell happens if you brings the O2 gases. So let's take a look. So I can use it here. So this might not be on the reading, guys. So I'm gonna be pretty quick with this, by the way. So basically, what will happen is, okay. So if you have a Rubisco enzyme, okay, which is the so Rubisco enzyme again is the enzyme that will fix the CO2, but again, instead, let's say, for example, it brings the O2 gases in. So basically, do you think they gotta produce? Absolutely not. It's not gonna happen. So it totally becomes a very wasteful reaction. Okay, not really a chain reaction, but the process of reaction that occurs, which occurs at the expense of ATP as well. Okay, so if I wrote it here, did I write the product here? Yeah. So basically, guys, what will happen is, so the first thing that this will create with the Rubisco enzyme, so it will add the RUBP. So this is a ribulose biphosphate with Rubisco. It produces something they call, guys, phosphoglycolate. Okay, so it doesn't produce phosphoglycerate. It produces what? Phosphoglycolate. Okay, which is obviously not what we're looking for. Okay. So what they do, guys, is so what you guys have to, what do you really have to know? You have to know what they do with this. Okay, so which means the purpose of this now is because they didn't get the right product. Okay, which means they know that they can't produce this one at all. Okay, so the purpose is at least get this back. That's the purpose. At least get the RUVP back. That's the purpose, okay? So what they do is they send this out, okay, to peroxisome, and also they send this to mitochondria, okay, for the conversion of whatever that was produced back into RUVP. So basically, guys, the peroxisome converts this into, if I'm not mistaken, glycine. Glycine is actually the amino acid. So phosphoglycolate, okay, which is produced in the chloroplast, they take it to the peroxisome. Okay, peroxisome has H2O2, right? And then they turn to glycine, which is amino acid. Okay, and then they send this out to, like mitochondria. Mitochondria converts this into serine, which is also one of the amino acids. Okay, and then at the expense of ATP, I think 2 ATP, it uses 2 ATP and try to put it back into RUBP. So it runs this cycle. Okay, this is what they call a photo respiration. So, which means like, because what? They bring in the O2 gas in there. So that's what they call a photo respiration process, okay? Which means this is a very wasteful reaction where they're using up the stuff they produce, which is ATP here, so they're losing it. So that's the reason why, guys, so there's a problem with Rubisco when temperature gets a little hotter, okay? And then, so the question for the plants is, like, how do they fix it? Like, so, like, what do they do, So do they stay like this? So when temperature gets a little hotter, so when the plant grows in a hotter environment, what happens, guys? Yeah, dude, they're gonna constantly have this problem. So, they're gonna constantly have to run this process instead of producing glucose, okay? So then what will plants do? Yeah, what will plants do, guys? So if they're not outputting enough G3P, then how they expect it, right? Like, oh yeah, we expect to produce like this many G3P, but we produce like half of it. Then what's the expectation for them? What do you think? They're gonna expect to what, guys? Dude, they're gonna, so plants, so this is one of the things we have to discuss down the road, okay? Plants, the photosynthesis occurs a lot on the leaves, right? On the bottom of the leaf, there's an opening. Okay, opening is called a stomata, or stomata, whatever you call it. Okay? So this is the opening where they bring out what, like O2 gases, and what, bring in a CO2 gas. Okay, and also what, H2O leaves as well. Okay, so they have to keep open the stomate in order to what, have the CO2 come in. Okay, but the problem is what, guys? If they leave it open, what happens, guys? The H2O will continuously leave the cell, which means what happens, guys? The problem is the plants are like, what if they open the stomate constantly? What happens? They're gonna lose water. So water is a very crucial thing for the plants because what? We talked about it, guys. Plants initially. So if you've done the reading, guys, like they kind of mentioned, I think, like 450 million years ago, that's when the plants came to life. Okay, so they're very sensitive about water. Okay, plants want to conserve water as much as they can. Okay, so because of this process, they constantly open the stomach. What happens, guys? Yeah, they're losing, because of CO2 not coming in. What happens, guys? They're losing water. So if the plants lose water, what happens, guys? It's gonna dry out, they're gonna die. Okay, so basically this happening with the photorespiration, the plants are what? Could potentially die. Okay, because why? Yeah, they're doing very wasteful product. Producing wasteful product by wasteful reaction. There's absolutely no benefit for the plants for doing this. Okay, maybe, I don't know, getting some amino acid, maybe, I don't know. So the good question to ask yourself is, where do they get the nitrogen from? Like what? Where did that get from? Like nitrogen? Like where did that come from? That's a question for a different day, by the way. Okay. So guys, so basically, guys, to kind of make this to make this process a little better, I guess, and to So the plants, they can only do this, right? Those are called C3 plants, by the way, C3 plants. Okay, C3 plants, they can only do this. They only have the enzyme that's exposed to the outside, okay, produce, okay, the Rubisco is gonna fix CO2, put it onto ribulose biphosphate, and what? Produce 3-phosphoglycerate, okay? What do they call C3 plant? Because what? Phosphoglycerate, a 3-carbon molecule. That's the first product created, which is a 3-carbon molecule. That's why they call it C3 plant. So there are different plants, okay, the plants that doesn't suffer from photorespiration, okay? And those are some examples. Those are something we call C4 plant, okay? C4 plant, not C3, but C4 plant. So what is C4 plant? So C4 plant is this. So maybe understand, guys, what a C4 plant is. So C4 plant, guys, is an evolution, okay? So again, the plants evolved, okay, to be a little more efficient with the carbon fixation process, okay? So basically what will happen is, okay, the plant cell, okay, plant tissue per se. Tissue is a group of cells that work together. So, so basically what happens is, so let me kind of give you understanding. So, so if you look at this, guys, I mean, so giving understanding, if you do the reading, they have a similar picture like this, guys, by the way. So the outer portion, this is the outside, okay? This is inside the tissue of the leaf of the plant, right? This is the mesophyll layer, the mesophyll layer of the plant, okay? And they call this portion what they call bundle, okay, of sheath portion of the plant, okay? And then inside is what we call the vascular, okay, vascular tissues, okay, which carries the water and also carries the product produced from the photosynthesis, okay? It's called a vascular tissue. So basically what will happen is, okay, they partition the job, okay? So mesophyll cells, okay, will fix a CO2 with a different enzyme, okay? Not Rubisco, different enzyme, okay? And then once they fix it, they bring it to the cells of the bundle sheet where they're gonna run the Calvin cycle. So the bundle sheet cells where they run the Calvin cycle is not exposed to surroundings, okay? Which means they're not exposed to carbon dioxide gases or even the O2 gases, okay? Which means what? Yeah, the mesophyll cells will bring in the CO2, okay? Will deliver the CO2, okay? So instead of like, them try to catch the CO2, what happens, guys? It'll be served on a silver platter by the mesophyll cells here. So that's what happens here, okay? That's what C4 plant structure is. Let's talk about how they do it. You have to know this, by the way. You have to know exactly what happens in the C4, the mesophyll cells and bundle sheet cells. So, so you look at it, guys. I'm going to draw two cells. So this is the mesophyll cell. Okay, this is the bundle cell. So I mean, this is a typical Calvin cycle. Okay, this is where the typical Calvin cycle occurs. Okay, where they bring us CO2, right? So how does the mesophyll cell helps them to do this? Okay, so they have a different enzyme. Okay, it's called PP carboxylase. Okay, that's a different enzyme. So they have a different cycle here. So PP. So instead of like here like ribulose biphosphate, they have a different what's called a PP. Okay, what is a PP, guys? A PP what? Phosphoenol pyruvate. Okay, so this is I'm sorry, phosphoenol pyruvate. Yeah, so where have you seen this? Yeah, it wasn't us. Where? Yeah, where have you seen this, by the way? Phosphoenol pyruvate? Where? Anyone? Anybody doing? Yeah, phosphoenol pyruvate. Where did you hear this? Where have you seen this? Oh, yeah, what are you doing?
The process with the COA into.
What?
Actually, never mind. I don't know. I remember seeing the reading, but I forgot what it was exactly.
What do you think?
About.
Isn't pyruvate the end product of glycolysis?
So, yes, pyruvate is in product, okay? But where have you heard like phosphoenol pyruvate? Where? This is what? This is also in glycolysis process, okay? This is a step like just before the pyruvate production, okay? So they take the phosphate out, okay? So what I'm saying is, okay, like every one of these guys are what? Like you've heard them, you've seen them, whatever, okay? So PP. So PP is what? Again, a phosphoenol pyruvate, okay? Which is what we know as well. It's a 3-carbon molecule. Okay. PP carboxylase, they bring in a CO2, okay, they fix the CO2 onto PP, okay, and they turn into what, guys? Oxalate. Yeah, did I turn it? I'm sorry, oxalic acetate. Yeah, oxaloacetate. Where have you seen this? Where have you seen oxaloacetate, Andy?
Citric acid.
Because where in citric acid cycle?
Like the last step.
Last step?
Before like the acetyl-CoA comes in again.
So acetyl-CoA, okay, will bind to oxaloacetate and what? Turns into citric acid. Okay, so oxaloacetate is what? Yeah, it is where the beginning. So yeah, you can say the beginning or the end of what? The citric acid cycle. So this being a 4-carbon molecule, okay, that's why they call C4 plant. Okay, CO2 added to a PP turns to oxaloacetate. This is a carbon 4-carbon molecule. That's what they call this what? C4 plant. Okay, whoever that can do this is what they call, guys, what? C4 plant. So PP carboxylate has absolutely no attraction for the O2. So O2 gases, okay, has absolutely no affinity for, or PP carboxylate doesn't have the affinity for the O2 gases. They have absolutely no attraction at all. Okay. And then oxaloacetate will turn into malate. Okay, so malate, yeah, so like, do you see this, man? Like, yeah, where have you heard malate? Where? Also, citric acid cycle. Okay, so like, this is like the thing, guys. Like, oh man, like, I've seen them, I heard them, right? It's like kind of going back a little bit, right? And what? This will be unloading the CO2, okay, into the bundle sheath where the rubisco can catch it and what? It turns into the 3-phosphoglycerate. And what happens, guys? This itself will turn now into a pyruvate. Okay, so malate gives off a CO2 and turns to pyruvate. And this, I mean, so how do you turn a pyruvate into a PEP, guys? Yeah, so how do you turn a pyruvate into PEP, guys? You need what, guys? They need ATP for this. Okay, the phosphate, they need ATP. You're going back the process of the glycolysis, okay? So at the expense of ATP, okay, turns it back to PP. So this process, okay, C4 plants, okay, where they deliver the carbon dioxide to the bundle sheet cell where they actually do the Calvin cycle, they deliver the CO2 in a silver platter, okay? But it's at a cost. What kind of cost, guys? ATP, okay? They need to use an ATP for this process, okay? But for sure, what, guys? O2 will not be coming in and what? Photoexpression can be prevented. So who's gonna run this process? Like, so where, in what environment is this will benefit, guys? Where, yeah, where the water is really lacking, okay, and also what? Temperature is relatively hot, okay? So what plants are considered C4, guys? Corn. Corn plants, okay, are considered C4 plants, okay? Keep that in mind. You gotta know, yeah, so even for the homework that you're gonna get, you have to know the steps here. Yeah, like the steps are, like, you gotta know your steps, guys, otherwise you're gonna have a hard time, guys. By the way. They're gonna ask you, like, where have you seen like this process? Okay, it makes what, like the reverse process of the energy payoff for the glycolysis. They're gonna ask that question, okay? So this is not something that your teacher will teach you guys on AP Bio. AP Bio, they don't talk about this. Okay, they don't. I mean, they talk about it, but not into a detail. Okay, I don't know, man. Like, you gotta know this for the Olympiad. This is easy stuff. I mean, you just have to like super easy. I mean, but if you don't know it, you can't like. It's not something you can figure out on the fly. Okay. So there's also, guys, something called a cam plant. It stands for something. I mean, you look it up, guys, on the picture of a cam plant. Okay. So example of cam plant, guys, is like cactus, even a pineapple. Those are examples of cam plants. Okay. So cam plants, okay. So these guys are, like, as you see them, guys. I mean, they grow like in a cactus, like they live in a freaking desert. Okay. What they do is, okay, the process is the same. Okay. They're similar to C4 plant. The process is all the same, guys. The difference is they opened their stomate. So bringing in the CO2. It occurs as a stomate, right? So they open their stomate only in the night. Only at night they open the stomate, okay? Because why? Like in the daytime it's too hot. If they open the stomate at the daytime, they lose all the water. So which means like they evolved to become what, guys? Only open a stomate during the night, okay, and store the CO2, okay, store the CO2, and when the sun becomes available where they can produce some ATPs and ADPHs, then they run what, the Calvin cycle, when the other resources available for them to run the process, okay? There's a couple things you have to know. So we talked about, guys, Calvin cycle, we talked about the C4 plants in terms of the PEP carboxylase and also the CAN plant. Also, we talked about photorespiration process as well. So let's talk about, guys, what happens in terms of how they produce an ATP. So how, like, okay, so the question becomes, how do they produce ATP? Okay, how do they produce an ADPH? That's basically what we'll talk about now. Okay, so like, okay, so we get it. Okay, so they run this process, produce P3P. Okay, and P3P can be easily turned into glucose. Okay, or can be turned into what, like fatty acid, like, you know, whatever. But now how do they produce the ATP and an ADPH? That's for the discussion now. All right, so let's just take a look. So, as per our discussion, right, this is a chloroplast, right, membrane bound organelles. And now they have these little, like, you know, stacks of what they call thylakoids. These are called, guys, thylakoids. And again, just so you know, okay, where that the Calvin cycle occurs is called stroma, because stroma is where the Calvin cycle occurs, right? This is where the Calvin cycle happens. Okay. So now the question becomes, guys, again, where, and then keep in mind, guys, the whole thing is called grana, by the way, which is made up of the sacks of little sacks of thylakoids. The question becomes, guys, where do they run? Okay, the process of the production of ATP, right, and also the electron as well. So the process occurs at the thylakoid membrane. Thylakoid. This is when they produce, where they produce the ATP. Okay. So let's take a look. So we have to kind of zoom into the thylakoid membrane. This is membrane bound organelle. Yeah. This is also have membrane there. So kind of make it a little simple here, guys. By the way, so we have. So these are, I mean, I'm sure you've probably seen this, guys, as photosystem 2, okay? And this is a photosystem 1. Okay. PS2, PS1, okay? Sometimes called this PS700. Very simple stuff, okay? Let's call this PS680. I might have swapped it, I don't care, whatever, okay? Like this might be 680, that might be 7, I'm not, whatever, okay? So, so basically what happens is, guys, here, so within the photosystem, okay, they have light harvesting proteins and also the pigments, okay, where they can capture the sunlight. Okay, that's basically what happens, guys, in the photosystem. So, so what will happen is, guys, okay, so the PS700, right? So this may be really good at what? Absorbing the sunlight at the wavelength of 700 nanometers. So this photosystem, guys, they have a pretty good job. They do a pretty good job of what? Absorbing the wavelength of 700 nanometers. 700 nanometers is what, guys? It's a red light. It's a red color. Okay. So a lot of these pigments are in the photosystem. They absorb red light pretty well. So these photosystem, guys, they absorb not only red, but they also absorb blue light and also the red light. So red and blue gets absorbed by the photosystem for the plants typically, right? So if you look at spectrum of the light, right? The wave spectrum for the visible light spectrum wise, guys, 700 nanometers is a red light, okay? And now this is basically, I think, like around 480 nanometers. That's the violet or blue, somewhere on there, guys. That's the lowest that you can possibly get. And then here is UV, and this is infrared. So where they get to absorb the most is what, guys? Yeah, somewhere here. So the absorption, guys, gets absorbed here and also here. So middle, they don't absorb at all. This is where the color green is at. Because they don't absorb the green light, the plants appear green because what? they don't absorb the green light. They only absorb blue light and red light. Okay. So what in the photosystem absorb these waves of the visible light spectrum is what they call, guys, chlorophyll. Okay, chlorophyll A, there's a B. Okay. One thing that matters for us. So within the photosystem, right? They have these pigments called chlorophylls. So chlorophyll has they have a sections of the chlorophyll, okay, where if you look at it, so they have keep in mind, guys, chlorophyll has a magnesium center, okay, and it has like pretty complex structure, okay, it has some nitrogens on them and stuff like that, guys. But just letting you know, guys, chlorophyll structure is very similar to the hemoglobin, by the way. Hemoglobin with the iron in it, guys, by the way. They're very similar, and they have a long tail of carbons, hydrocarbon tail. Okay, so that's what the chlorophyll looks like. So one of the stuff they've talked about is because it has a metal center, what happens, guys, they're pretty good at what, guys? Displaying the electron, displacing the electron, okay? So they have electrons, and what? If sunlight hits it, what happens, guys? Yeah, it's what? Electron can be ejected from chlorophyll itself, okay? So I think I wrote it something here. So I think they have a name for this, guys. Like I think they put here porphyrin, like this portion, and here's a hydrocarbon tail. Okay, hydrocarbon tail. So basically, guys. I mean, so this they have a name for this portion. They have a name for this portion, guys. And it's in the reading, guys. Please read it, please. Okay. A and B, guys. I mean, they have a very similar structure, by the way. Okay. They're all exactly about the same. Okay. But their wavelength they absorb is just slightly different, by the way. Okay. The wavelength of absorption, where the maximum absorption occurs, is just slightly different, guys. Okay. And there's also, guys, one other pigment, okay, carotenoids. This is like, like they absorb a different band. So their color appears to be more yellowish, like carrots, right? Like carrot, like orange. Okay. I mean carotenoids. So if you look at it, guys, so this guy is also one of the pigmentation as well. Okay? They all be found in what? In the photosystem. Okay. So these are what? Their photosystem is there for what? Light harvesting. They are there to absorb the sunlight. Okay. So so guys, I mean, so if you read this, guys, I mean, if you read the chapter, guys, but they explain it like pretty, like, very simple, guys. Nothing really crazy, nothing fancy, guys, by the way. So basically what happens is the light gets absorbed, okay, and then within the photosystem, okay, they have something called, guys, a reaction center. Okay, they have something called a reaction center in the photosystem, okay. And then within the reaction center, they have a chlorophyll A. Okay, they got chlorophyll A in there, guys, by the way. Yeah, chlorophyll A is we found the reaction center. So the sunlight that was absorbed, okay, what will happen is this will what? Excite the electron, because electron excitation in the reaction center. So they have all these, like, other pigments here, guys, which are system, providing the energy for the reaction center to excite the electron, okay? The electron exciting is what? They're taking the electron away from the chlorophyll, okay? So the reaction center, chlorophyll A, is losing an electron. That's what's happening, okay? And now there's an electron acceptor, okay? So in the reaction center, guys, they have an electron acceptor of the electron. So I mean, again, you know where this is going, guys. The acceptor of electron, guys, what happens, guys? Electron now is being excited, okay? The acceptor accepts the electron for what, guys? Which means it'll run what they call, again, electron transport chain, okay? So you got to understand, guys, here. So so one of the stuff that we talked about is the photosystem, okay? They lost an electron here. The electron popped out, okay? Which is excited by the sunlight, okay? But now the question is, guys, okay? There is a limited number of the photosystem, okay? Which means they have to replenish the electron, okay? So where are they going to get the electron from as a replenishment perspective? So like, okay, so they're losing electron, okay? Who's gonna give them the electron? That's what's gonna happen, right? Like, where are they gonna get it, right? So that's a question to ask yourself. So what we talked about was what, guys? Water, H2O. So water is what? Water is the one that ultimately what, guys? Ultimately sort of what? Replenishes the electron, okay, for the photosystem 2. So the reaction was what? So I wrote it down last week, right? H2O turns into what, guys? H plus electron and O2 gases here. So this will turn into this, and what? Electron that was produced by this will replenish the electron that was lost in the photosystem 2 in chlorophyll A. If you look at it here, guys, O2 gas was produced. You know what's an O2 gas, guys? I mean, so O2 gas is not something they would use, okay, in the chloroplast, right? So this will be leaving the chloroplast, okay? So what do you call something like this in bio, guys? They call this what? Byproduct. They call this byproduct, which is what? This is a product where they don't really need this, which means that they're gonna take this out, okay, and get rid of. So I mean, so O2 gas can be used by what, guys? Like mitochondria. Mitochondria can take this and what? Use them as a lost electron acceptor for their electron transfer chain, for example, okay? So now the question becomes, guys, is electron here. So they can run the electron transfer chain. So, you know, they're very similar, guys, just so you know, in terms of what they got. They got a cytochrome. Okay, of course, they got cytochrome, right? The cytochrome, which can what, guys? So it's the difference is electron transfer chain, the understanding is the same as a mitochondrial electron transfer chain, right? Difference is what, guys? They actually pump in the H plus. So if you recall, guys, okay, so mitochondrial electron transfer chain was what, guys? They pump out the H plus, which is proton, into the intermembrane spacing, okay? But now this is kind of the other way, right? Where the electron transfer chain of the chloroplast of the this is called the light-dependent reaction. What? They pump in the H pluses instead of pumping it out. So, and these are called the proteins, electron acceptors. By the way, this is I think it stands for PQ. This is a PC, I think. I think this is plastoquinone. This is a plastocyanide. Okay, that's the name of those guys, I think. So, like ubiquinone, guys, of the mitochondria. This is a plastoquinone. So you gotta know these names, like details, okay? So, dude, it doesn't matter if I write it, guys. You gotta memorize this. I mean, this is the freaking reading, guys, okay? Plastoquinone, and this is a plastocyanide, okay? So they are assisting of what? Moving the electron from one to the other. So again, you know where this is going, right? So it produces what, guys? So concentration gradient of H plus, constant, like, you know, electrochemical gradient of H plus inside the thylakoid, okay? Which will what? They will use ATP synthase for this. But now, let's take a look at this, okay? So now the photosystem 1. So let's not even go here. So photosystem 1, right? Sunlight will hit it. Okay, the same process, guys. They have chlorophyll A, electron rejected, by the way. So basically what will happen is electron gets excited. Again, there's an electron acceptor here, okay, which can run the electron transfer chain again, okay? This can run ETC. But this ETC is a little different, guys, by the way. Okay? This ETC, guys, is a little different because this ETC does not pump in the H pluses or anything like that, guys, by the way. What will happen is, guys, so they have a protein called the FD, and also a protein called, guys, I think they have a protein called NADP plus reductase, if I'm not mistaken. Okay. So, which means, guys, these guys do not, okay, so for the second photosystem, right? So they do not pump H pluses, okay? So the electron will follow the direction, okay? This is the ferrodioxin, if I'm not mistaken, guys, by the way. So it just passed the electron down. And this is an NADP plus reductase. So the name kind of tells it all. What happens, guys? What do you think this guy will do? This guy will do what? Reduce NADP plus and H plus into what, guys? NADPH. That's what's happening. So basically, guys, what happens is when it gets to this enzyme, so this electron is a high energy electron, right? Basically, what will happen is NADP plus and H plus and 2 electrons will turn into what, guys? NADPH. So this is where they produce NADPH. Okay. This is what high energy electron will be going down to the Calvin cycle, which helps them to assist to reduce 1,3, okay, biphosphoglycerate into G3P. So, like, you guys have to, like, dude, like, you guys have to be, like, pretty good at this, guy. Like, this is what I'm saying. Do you recall, like, dude, like, G3P, okay, into what, guys? 1,3, okay, biphosphate, glycerate, and also it turns into what, guys? There's kinase and what, like 3 phosphoglycerate. Okay, so like that's basically what happened, guys. Okay, during the glycolysis process, right? This is the energy payoff. Okay, so they take the ATP, take the phosphate, and produce ATP here. So what we talked about is what, guys? The Krebs, the Calvin cycle is what, guys? The opposite direction. So if you recall, guys, okay, this is where they what? NAD plus will turn to NADH. Yeah, so this to this, guys, is where G3P to the biphosphate, right? This is where they oxidized. Dude, like, so, dude, like, this is G3P to this is an oxidation process, right? This is a process of the glycolysis, right? G3P to 1,3 biphosphate glycerate is what, guys? That's an oxidation process. Okay. Keep in mind, guys, what they're doing is what, guys? They're going back this way. Okay. Which means what, guys? They have to what? They have to reduce. They have to reduce. What are they going to reduce with? Electron. And who will supply the electron, guys? NADPH. Okay. And that's what we talked about during the Calvin cycle. Okay. So they need to have a higher electron. So from PS1, okay, which means what? Yeah, there's an enzyme, NADP plus reductase, where they produce what? NADPH, where it has the electron, where it's going to carry down to the Calvin cycle and what? Put it in there to what? For the reduction of the 1,3 biphosphate glycerate into what, guys? Okay. But as you guys remember, guys, okay? This process of the Calvin cycle we call what, guys? Yeah, 9 ATP and what? 6 NADPH. Okay? And also in case there was a photorespiration, okay, they need more ATP, right? So what are they gonna do, guys? So if they need more ATP, what happens, guys? Okay, so basically how this ran, okay, so look at this, guys, okay, electron gets excited, it runs down, produces some ATP, right? There's ATP synthase. So we didn't get to that part yet, but and now keep in mind, guys, here, yeah, so now, yeah, look at this, guys, okay? So photosystem 2, okay, what the electron lost by this was replenished by what? Water. What about this guy? Photosystem 1, okay, lost electron also, right? Where did they get it from? Where did they get to replenish electron from? Is what? Electron came down on the ETC. So from the photosystem 2, where the electron is running down, it actually replenished what, guys? The photosystem 2 electron. Okay? So there's water doesn't get involved here. Okay? But now they run it down. Okay? So this is where, if it runs like this, that's what they call, guys, a non-cyclic. You know, like they call this, guys, it produces ATP, right? They call this guy non-cyclic photophosphorylation. So remember, guys, what do you call that, guys? Mitochondria, right? Mitochondria, how do they produce ATP, guys? What do they say? Oh yeah, it's called the oxidative phosphorylation, right? Because why? It's a constant oxidation reduction reaction, okay? Like as the electron transport chain goes, right? They call this process right now, as it stands right now, is a non-cyclic, okay? Photo, which is light, right? Phosphorylation. So, like the way they produce ATP, same, because they got ATP synthase. Okay? They got, like, look at it, guys. I mean, they have an ATP synthase here, okay? ATP synthase, same ATP synthase. Okay, there's no difference between this. Okay, they have the F0, F1. The H plus pops back out. It turns the turbine. You know, it activates the F1, the portion F1 portion of the ATP synthase. It was able to catalyze ATP from ADP. It's the same process. There's nothing. So we're not talking about this again. Okay, the same process again. But what if they need more ATP? I mean, they don't need more NADP. H, right? But what if they need more ATP? What do they do? That's when they call what? Make this into a cyclic. They can turn into a cyclic phosphorylation process, okay, instead of a non-cyclic. So the electron gets excited by the PS1, okay? They can put the electron back into the cytochrome for what? Pumping out the H pluses, okay? So this process right here, okay, that's called a cyclic, okay, phosphorylation process, okay? So which means what? Yeah, so the electron travel down, it bounces back. Instead of going to non-cyclic process, they can what? Turn it back into the cytochrome and what? To pump out more H pluses, okay? Which to make them into what, guys? A cyclic process, right? Very, very simple. It's nothing fancy, okay? So yeah, that's how they produce, guys, ATP. That's how they produce, you know, the NADPH, which high energy electron, okay? Somebody else. Question concerns? Let me know. Yeah.
So does cyclic not produce NADPH?
Hold on. If you go here, it goes down this way, and if they produce enough ATP, what happens? They go down this way. Questions, guys? Any questions so far? Let me know. Okay. So let's talk about this, guys, okay? So production of ATP. You put it this way, right? What is different with this in comparison to the production of ATP for mitochondria? So so they produce ATP, okay? This guy produces ATP, okay? What is different with the production of ATP by phosphorylation versus the mitochondria, okay, where they call it oxidative phosphorylation process? What do you think? What's something that pops up to you guys that's something that's really different about this? Is what? Where they get the electron, okay? So where the electron comes down, where the electron is gained is a little different, okay? So electron is found in, okay, the pigments where they absorb the electrons, absorb the sunlight, right? And also so basically, guys, just so you know, as we understand, right? Just so you know, for the mitochondria, it was like chemical energy, which is of the glucose, okay? The potential energy of electron, like chemical energy, okay? Energy in the glucose, right? It was converted to what? Electrochemical gradient, right? Electrochemical gradient of H plus, okay? And energy using the electrochemical gradient, guys. So yeah, like they turn into NAD, NADH and stuff, guys. And what? This is how they produce the ATP, right? But it's a little different for photosynthesis here, because why? They actually use a sunlight here. So the energy they get is what? Sunlight. Okay. Sunlight is the source of energy overall. That's the ultimate source of energy for this process. Sunlight will excite the electron. Electron gets excited, okay? And they don't need what? I mean, they don't need this. So sunlight will be what? Sunlight directly can provide what? The electrochemical gradient of H plus, okay? And then, like, yeah, down the road, what happens, guys? Electron that was displaced is what? Will be utilized and captured by NADP plus. Okay, that's pretty different here, okay? Like, it was a little different, guys, because the mitochondria, they produce NADH during the process. So Krebs cycle and glycolysis, the main process of those is what? For the production of NADH, okay, which is it captures a high energy electron and unload the electron for ETC. That was what they call the ETC for the mitochondrial electron transfer chain, right? It's a little different because what? Yeah, like, dude, it's kind of the opposite, right? NADPH gets produced at the end of the process. So if you think about it, right? the last electron acceptor is what? Is this? You know what I'm saying? So the last electron acceptor of the mitochondria is O2 gases. Okay, O2 gas, electron in H plus turns into H2O. Okay, but now this is like somewhat kind of going backward. Okay, so H2O turns into H plus and electrons in O2, and then the electron went down, and what happens, guys? And what? Reduced in ADP plus. It's sort of the backward way if you think about it a little bit. Okay. So ultimately, the way they produce the ATP is the same. Okay, they produce electrochemical gradients. Okay, and then what? Use the electrochemical motive force. Okay, the proton motive force to produce ATP. Okay, but there's a slight difference in terms of how they execute the process. Any questions? Let me know. And keep in mind, guys, photosynthesis, okay, can be performed by plants, right? Plants can do this, right? Keep in mind, guys, there are some prokaryotes can do this as well, okay? Some of the bacteria can what? Perform this as well, okay? And also, there are some eukaryotic cells also as well, which is not a plant, that can do this also as well, okay? So photosynthesis is not only done by plants. Okay? There's other organisms that can do this, which also can be just what? They can be just prokaryotic cells, by the way. Okay? Any questions? Okay, so guys, take your homework out, guys, which is the. I mean, if we finish this earlier, which is, I'm kind of shocked, but then I'll move on to the next topic, which is communication. But can you go back? Can you go to your homework, guys, and get ready, guys? Let me get some water. So I'm not gonna record, guys, the homework review, guys. Okay?